we're going to start with our warm-up. So let's do with that. Give me a few. All right. And like I said, you should still always look, the 1 point isn't going to make a difference. But if you're finding like you did the warm up and you just ate it, that is a good, it's kind of a red flag that this is material you got to review, because like I said, everything keeps building. Name at least 2 different ways that ion channels. So I haven't finished grading all of these yet, but most people got this. You know, what are the 3 ways that we talked about that ion channels can be controlled, can be gated?
Voltage, ligand, and mechanical.
Exactly. There was voltage, just changes in the membrane voltage or membrane potential, electrical potential can be the signal for a channel to open or close. That's going to be really important when we look at excitable cells and action potentials and things like that. Ligand gated, where there's some chemical messenger that controls the opening and closing. You know, that's how neurotransmitters excite a neuron, or hormones can talk to a cell. That's again how like your taste buds work. I mentioned like sugar can open up ion channels that creates electrical signal that makes your brain know that your taste buds are noticing sweetness. And then the mechanically gated, just physical deformation, stretching of the membrane can open channels, create electrical signals. You know, that's how your sense of touch works. That's how your sense, even like I said, hearing, which is just vibration of your eardrum. So we'll be seeing lots more. In fact, later today, we're also going to get deeper into how is that connection happening, like particularly for ligand gating. So some signal molecule comes in. What are the steps in between the signal molecule coming in and a channel opening? You know, it's not just kind of magic. There's some physical chain of events that has to happen. So we're going to talk more about that, what we call signal transduction. All right, cell organelles that are primary, that are the site of protein assembly. So this one, I gave credit for either ribosomes. Ribosomes was the main one that I wanted, because that's the kind of core place where amino acids are being assembled. If people put rough endoplasmic reticulum, I gave you credit for that as well, because you know it's rough because it's studded with ribosomes and it's where proteins are getting made and just kind of getting threaded directly into the ER. A lot of people wrote Golgi apparatus, and that isn't really the right answer. That is kind of more where things are modified. You can add extra groups to things, you can store and transport and move things around. But Golgi is more about like kind of aftermarket modifications, so to speak, right? The proteins actually get made at the ribosomes.
I think where we were confused with the Golgi apparatus is you really said that it was where proteins are assembled, packaged, and completed. So maybe
I said like more for like you know, for like the glycoproteins and stuff where you have to do lots of extra stuff on them.
Because underneath the rough endoplasmic reticulum, it says only protein synthesis and assembling and storing protein. Like I wrote word for word what you wrote.
I have the exact same thing. I had the Golgi apparatus as like the pancakes and its protein assembly, packaging, and moving things post-processing.
So if I said the actual assembling of the proteins does not occur there. I mean, this is something, like if I said that, then that was unclear. I did it unclearly. The actual assembling, actually putting the amino acids together with peptide bonds, that happens at a ribosome. So it could be at a free ribosome. It can be at a ribosome that's anchored, like on the rough ER. But you should know that the actual assembly of amino acids into proteins is at a ribosome. So this will be the clarifying moment. So if this shows up again,
So do we get the point since we
It-
totally had the wrong information?
the point doesn't matter, right? The point, if you want, I can change that or something, but it really is a fraction of a fraction of a point. So I wouldn't worry too much about it. It would be like a grain of sand on the beach. Let's see. This one, a lot of people did not get this, and this is going to be super important for you to realize or understand the idea of how membrane voltage creates forces on ions. In fact, maybe I should do a quick little.
I thought when it was negative 70 MV, it was like homeostasis, right? Like they stayed.
No. I said that that's often the resting potential for excitable cells, but it's a, here, hold on. So if I have some cell and I look at the voltage across the membrane and it's minus 70 millivolts, that means that the inside is kind of more negative compared to the outside by a certain amount. Because we always talked about the voltage being the inside with respect to the outside. So again, a typical neuron or muscle cell tends to, at rest, if it's not getting excited, kind of equilibrates around here, but we'll see why in a little later today, actually. And so given that we have this situation where it's negative inside with respect to outside, and then I look at some cation, maybe sodium. What is going to happen to this? What's it going to want to do?
Move inside.
Inside.
It's
Gonna
attracted,
wanna
so it's
move
going to
in.
want to move.
Exactly. It is positive, so it's going to want to move towards the more negative. So there's going to be an electrical force drawing this cation into the cell due to the negative voltage across the membrane.
What then is the significance of knowing that cells rest at negative 70? I think that's what confused a lot of us.
Yeah, it's going to be really important when we talk about voltage-gated channels, for instance. Voltage-gated channels that only open at minus 55 are going to be closed when the cell's at rest. And it's going to be able to let us know if a cell's excited when the membrane potential is starting to rise and become positive, for instance. That's going to be during like action potentials, and that's when like neurons are sending electrical signals down their axons, for instance. So the resting potential is, it's kind of like, your resting temperature is like, 97 degrees. And if your temperature is 104, I know you got a fever. Or if your temperature is 60, I know you're hypothermic. So it kind of just gives you what is the normal thing when you just look at it, when everything's kind of resting, and then we can then notice when things are in different situations.
Would it be fair to say that at its resting state, it's kind of, you were saying equilibrium before, which I know that's how the cell wants to be with that negative 70 millivolts on the inside. But if all the doors were open, it would
Right.
equilibrate, and it would eventually, the charges
Right.
would
Yeah,
balance
but
out.
we're
So would
gonna
it be fair
find-
to say that like it stays that way to be primed for an event, to be ready?
I would say on a practical level, yes, that's totally true. And we're going to talk about what the equilibrium at minus, the reason this exists, the reason it exists is because of a combination of both what kind of channels are open, what's the permeability of the membrane at rest, and also what are the concentrations of sodium and potassium inside and out of the cell at rest. So, I mean, this number comes from a variety of conditions that exist, which we're going to talk about today. We're going to talk about concentrations of sodium inside and outside the cell, intracellular versus extracellular fluid. We're going to talk about what are the normal permeabilities of the membrane when a cell is at rest. And we'll also talk about what happens to those permeabilities. How do they change when the cell gets excited? We're going to see there's a big shift actually from primarily potassium permeability to sodium permeability, which is going to radically change the equilibrium potential that things are going to run to. So it'll all become clearer as we go deeper into this as today unfolds. So, but this minus 70 millivolts, where the a normal excitable cell tends to be at rest, that is due to lots of conditions we haven't talked about yet. You know, like kind of the normal, the kind of the resting permeabilities, the typical concentrations of ions around, and it's going to be a mix of diffusion gradients and electrical gradients that are going to ultimately have to find balances to create these equilibriums. So again, we're going to see all this in more detail. So now I'm going to go back and we will continue over here. All right, so hopefully that's clearer now, that if you had a minus 70 millivolts membrane voltage, a cation wants to come in, where it's more negative. If I had a membrane potential of plus 50 millivolts, what would a cation want to do?
Move out.
Move out.
Exactly, want to move out because it's more negative outside. Yeah, so think about it like that. And then as we continue on, this stuff's going to feel more intuitive.
Is there any time when it would like not do anything, oscillate and bounce in and out of the cell?
No, that doesn't make that. That was supposed to just be kind of a goofy answer. Yeah, I mean, if there was no potential at all and no diffusion gradient at all, then it would be kind of random whether or not it's moving one way or another. But so I guess that could be. But yeah, I was trying to come up with answers, because you want to have a variety of answers. So I was like, okay, what could I put there? Let's put one. That'd be kind of fun. Let's say it's going to bounce. Let's see. Choose all that are true about facilitated diffusion. All right, so this, again, we talked about, this is that special case of diffusion using carriers, but it's still diffusion. It is not active. We're going to talk about different forms of active transport today, but facilitated diffusion is still diffusion. It's still just going to go from high to low concentration. It's not going to require an addition of additional energy. Yeah, so it's a form of passive transport. It requires special membrane proteins called carriers. And then the big difference between just regular diffusion and facilitated diffusion was that there was a rate limit. I talked about the carriers being like a revolving door where there's just a maximum number of molecules that can move through this from one side to the other in any given amount of time. Because there's a whole kind of docking and conformational change and getting spit out the other side and stuff. So again, we'll be looking at these and more kind of fancier versions of carriers, these like couple transporters as soon as we're done going over the warm up. And finally, glycoprotein. Right? If you remember, glyco for sugar, like glycolysis is breakdown of glucose. So glyco, it's got sugar groups, and sugar is just a carbohydrate, and protein. So it's a combination, a hybrid between a carbohydrate and a protein. It's mainly protein with like these sugar groups, carbohydrate groups attached to it. So carbohydrate, protein. I mean, again, cholesterol is a lipid. Nucleic acid. Yeah, so again, glycoproteins are kind of the main things on the cell membrane, embedded in their membrane proteins that are used for like cell ID and things like that. We'll be talking more about that. In fact, I should mention a term. I think I put it in the study guide, and if I don't actually mention it in class, everyone's going to ask me, like, what is this thing on the study guide? On this idea of the Glycocalyx. Glyco sugar. Calyx means kind of like a husk, like a cover, outer coating. And if we look at a cell and we have all of these membrane proteins embedded in the membrane, most of these are glycoproteins. So they have these little sugar groups. So if you are actually looking at the cell from the outside, this is supposed to be an eyeball. What you actually see are all these like kind of sugar groups. You know, people talk about the cell being kind of like sugar coated. So that's what the glycocalyx is. It's that kind of bunch of carbohydrate groups that are part of the glycoproteins that you actually encounter as you like look at the outside of a cell from all of these membrane proteins. And again, a lot of it, some of it has to do with kind of cell identification and things like that. But that's what this word glycocalyx means. It just means the sugar coating on the cell due to the carbohydrate groups on the glycoproteins that are these membrane proteins. Obviously, I'm playing fast and loose. Like a face this big would not be in the same scale as a cell like this. So this is some infant, this is, what is it, Maxwell's demon or something. All right. So last we left everything. We were starting to do kind of our list of membrane proteins. And we started with transport, and we were still in the middle of transport when we ran out of time. So we're gonna continue with transport and then talk about lots of other kinds of membrane proteins, and then see how they work together to do different things and get a little deeper as well. So here we go. So we talked about transport. There were ion channels. And these are often gated. So we talked about that quite a bit. There were carriers. These are usually for molecules that, polar molecules that wouldn't normally cross the membrane, such as sugars or amino acids and things like that. Still diffusion through them, but again, facilitated diffusion. So there's this limited rate. Then the place where I kind of left off was coupled transporters. This is what we're going to go into now. So these coporters. I think that's what they call them. The idea here is you've got like a membrane. And you've got your membrane protein, that is this coupled transporter. And it is going to only let things move across if two or more things move together. Like some of them are what we call coports. They're both going in at the same time. You can have, not a coport, I'm sorry, I'm spazzing on you here, symports. Symport is if they're both going in at the same time. In order for instance, we're going to see one where you can have glucose and sodium work together to go through this thing. But both the glucose and the sodium have to be docked in order for this thing to then spit them out on the other side. Another option is what we call an antiport, where one thing goes in and one thing goes out. We're going to see really important antiports, for instance, that are sodium leaves and potassium comes in. So antiport is one thing goes in and it's coupled to something else going out. Symports, two things have to move the same direction. They could be moving in or out, but they have to both be docked and both move in together as a, as a couple, so to speak. It would be like there's some party and no singles allowed. It's like if the glucose comes here, they'll say, I'm sorry, you're not allowed across. The sodium comes in alone, I'm sorry, you're not allowed across. But if they're both coming in together, it's like, all right, boom, they trigger this thing to then spit out on the other side.
Did you say just two things or two or more?
There's a whole variety of ones. The ones we're going to look at specifically tend to be more like the one. In fact, the one I just drew here is going to be important in transepithelial transport in a few moments. Antiports are also important. And again, one of the most important ones of these actually has multiple sodiums and potassiums that move together. So it's not just one ion on one side, it can be more than one.
So with the antiport, this is happening simultaneously. The calcium's moving in while the sodium's moving out, right?
So K, if you want to remember.
Or potassium, I'm sorry.
Yeah, calcium will be that. Yeah, so exactly. The potassium can only move in if a sodium is moving out at the same time. It would be like, it's kind of like, yeah, think about like there's some party that is at capacity and the fire marshal is like, no, nobody else comes in here. And so you're just kind of waiting at the door until somebody finally leaves. And then once they leave, you can come in. But you can only have a person come in if somebody's leaving at the exact same time, something like that, right?
You said that there could be multiple at once, right? Would
Yeah.
they have to have an equal number of ions on both sides or no?
No, it depends on the specific membrane protein. So in fact, yeah, the most important sodium, potassium antiport has like 3 of one and 2 of the other. So it's not. All right. So the other thing I'm going to add into our picture here is these can also so you can also have some of these more complicated transport proteins that are actually pumps that use the power of ATP to push things against their concentration gradient. So let's talk a bit about active transport. So let's start with what's called primary active transport. This is use ATP to directly provide energy to move something against its concentration gradient. So I'll give you a few examples. One of the main pumps in almost all your cells that actually are we're going to talk about the concentrations of ions inside and outside a cell, is the sodium potassium pump. Pumping potassium into the cell, pumping sodium out of the cell, even though there's already way more sodium outside, we push even more out. And there's already a lot of potassium inside, but we keep pushing it in. So potassium isn't going to want to come in. So in order to make this work, we have to drive it with the breakdown of taking ATP and breaking it into ADP plus phosphate, right? And that's also gives us energy. So we use this as kind of the fuel, kind of the gasoline, whatever, to run this little motor, which normally wouldn't go, because normally this, I've already got tons of sodium outside. We don't, sodium wants to come back in due to its diffusion gradient, but I can use the power of ATP to keep pushing it out of the cell, even though it doesn't want to move that way due to its diffusion gradient. So this is, moving, low to high concentration, right? Which is the opposite of diffusion. Diffusion, things move from higher to lower concentration. So primary active transport, there's typically some kind of membrane protein. We're going to see, like in the kidney tubules, we're going to have things that just pump sodium, and then again are using ATP to make that happen. Right, so you can have lots of different versions of these primary active transport things. Sometimes things just sometimes we're just moving one thing out according to its, against a concentration gradient. This sodium potassium pump is actually more complicated. It's moving sodium out and potassium into the cell. But this idea of primary active transport, use ATP directly to provide the energy. There's another kind of active transport which you need to know, which is called secondary active transport, which is actually going to use a lot of the things we've just been talking about. So secondary active transport. Should be either the second. Nabbit. So I'm gonna say it, and it's gonna sound weird, but then I'll explain it, and then it'll make more sense. So I'm gonna just lay it out first. This is using the energy stored in a concentration gradient. Usually that was initially created by primary active transport. So it uses this gradient, along with a coupled transporter, or I should say along with coupled transport. To move some, something against its concentration gradient. And it's all right. So this sounds all weird. So let me explain a little better. So let's say here's this is my cell. This is like the inside of the cell here. So I'm going to create a concentration gradient. Like I talked about that sodium pump. So I might have this thing here where I am pumping sodium out. So there's lots of sodium now outside. And this pump is being driven by ATP. So this is going to be an example of primary active transport. And it is setting up a concentration gradient. It's setting up a situation where there's a lot more sodium outside than inside. This looks like the Batman theme, right? Na 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 na. So we have lots of sodium outside of the cell here. And now there's a sodium concentration gradient. And I talked about in the very first week of class, one of the ways we store potential energy is in a concentration gradient. Again, I'm going to personify it, but sue me. Sodium wants to come back in. Like we got lots of sodium outside, not so much inside. It wants to come back in. And if you give it a chance, it will come back in. So we are going to use coupled transport. We're going to have, let's say this is in the intestine. Let's say we have I'm going to make it blue so it's a different color. Let's say there's glucose around here, and I want to bring the glucose into my cell. And there's already a lot of glucose in the cell because cells have glucose. What I can do is I can use a symport that couples the movement of glucose and sodium. Like sodium, it lets sodium in, but it only lets sodium in if glucose comes along with it. So what's going to happen is the sodium wants to come in, and it's going to just, I want to come in, and then a glucose bumps in and and binds in its little spot here, and bing, they get dumped onto the other side. So, again, the way to kind of think about it is the glucose is getting dragged in along with the sodium. The sodium wants to come in because there's this energy stored in the concentration gradient of glucose, of sodium, that was originally set up by active transport over on this side. But secondary active transport means the glucose is getting pulled in against its gradient, but the energy is not coming from ATP. The energy is coming from the energy stored in the sodium gradient. And the sodium gradient, which was originally set up by ATP, but the actual energy to move glucose into the cell here is coming from the energy stored in the sodium gradient. So does that make sense?
So this secondary active transport cannot happen without the primary active transport, correct?
In general, yeah, in general, or something has to set up the gradient. Sometimes we're going to see there can be other things that set up gradients, but typically this is the typical situation. This is a typical, something has to set up the concentration gradient. And then the energy stored in the concentration gradient is what drives the movement in secondary active transport.
Okay, thank you.
Since we're doing active transport, just for completeness, I'll add one extra piece into it. So active transport extended. I'm gonna need to slow down my writing here. So most of the active transport we'll be looking at in the class is going to be either primary, driven by ATP driving pumps, or secondary with concentration gradients using these like symports to help pull things. But another important thing that we'll also see that is active is endo and exocytosis. You want to get something in and out of a cell, particularly if it's a bigger thing. You don't drive it through a little membrane channel or carrier. Endo and exocytosis, which you all have seen. You have the cell, and let's say I have some little thing that I want to get into the cell. I pull. This would be, endocytosis. If I ran it this way, it would be exocytosis. I can take something that's in the cell and spit it out. So this is a way to get things across the membrane into the cell or out of the cell. And it's going to take energy. You're going to have all sorts of cytoskeletal machinery pushing and pulling on the membrane to make this happen. So this is a form of active transport because it's, it doesn't just happen on its own. There's a lot of energy involved in the machinery to make this happen. Some of the things, some words I should mention, the piece of membrane that is involved in this is not just some generic membrane. There's usually a piece, often what they call, like, there's this stuff called clathrin. It's another membrane protein. This is what we call a clathrin-coated pit. Clathrin is this membrane protein that is found specifically on the little parts of the membrane that are going to be involved in endo and exocytosis. It gives things for all the cytoskeletal stuff to kind of anchor and push and pull. And it also does this really cool thing where when this pinches off, the clathrin interlocks like a cage to stabilize a little vesicle in there. So it's not just some random little squishy thing of phospholipid. It's actually stabilized with this cage of protein that stabilizes the little vesicle. So clathrin is, just, you don't have to know beyond kind of understanding the way I mentioned it, but I just want you to think about endo and exocytosis as being a little more involved than just like some cartoon of some Pac-Man. Endo and exocytosis is also used to very quickly change like the permeability of a membrane, right? So another example here, I could have a cell that has a whole bunch of channels in it. Let's say these are channels to allow some ion or water or who knows what go across. But now the conditions change and now I want those out there. I don't want that permeability there. We're going to see this in the collecting duct in your nephrons, in your kidneys. You are going to want to sometimes have the collecting duct permeable to water, sometimes not. Sometimes you want these aquaporins there, sometimes you don't. And you want to have a pretty quick ability to add them in, take them away. If I have these membrane channels here and I want to get rid of them, all I need to do is endocytosis. So now the cell is no longer having these channels in the membrane, so it is no longer permeable to that stuff. And this happens quick. You can just pinch it off and suck it in. But we can do exocytosis. We can refuse this little vesicle back with the membrane, and almost an instant, we got the channels inserted back into the membrane. So endo and exocytosis are a way to really quickly and easily change the properties of your membrane by inserting proteins or pinching them out and taking them out of kind of active duty. So does that make sense? So, Natalia, you've got your hand up.
So I had a question about active transport. I just wanted to confirm that basically active transport cannot happen without any type of like concentration gradient prior to this happening. It doesn't necessarily have to be primary active transport creating the concentration gradient. That was my question.
For secondary.
Oh, yeah, sorry, that's what I meant.
Yeah, so secondary active transport is driven by a concentration gradient, which is typically set up by active transport, but there's probably going to be some examples where it's not exactly true.
Okay,
but
I just wanted to put it in my head that it wasn't always primary active transport. It could be other things as well. That was my question.
It's typically.
Okay. Okay, thank you.
Sure. All right, so yes, I think this is interesting to just think about. Endo and exocytosis can be about bringing things into a cell or out of a cell, but then it can also be about actually just changing your membrane properties quickly. Okay. One of the examples that I always use to try to put together a lot of these concepts, and that is going to be coming up in the digestive system and the urinary system and lots of different things, is the idea of transepithelial transport. You should definitely understand. I'm going to go through this, and when I'm done, you should definitely understand all the different parts and pieces. I promise this will be on your first exam in a couple of weeks. You know, that you really do need to understand this idea, what it is and how it works, because it's very common. The basic idea is epithelial tissues, epithelial sheets tend to form these boundaries between the insides and the outsides of things. So for our example, let's do an example in the small intestine. Anybody who's been in anatomy, what kind of tissue lines your small intestine?
Is it
Epithelium.
stratified squamous?
Not stratified squamous.
Simple.
Simple.
What's that?
Oh no, columnar?
Yeah,
Yes.
simple columnar.
Simple columnar. So we have simple columnar epithelium. So we have these kind of columnar looking cells. So this is just supposed to be the simple columnar epithelium of my intestinal lining. Up here, this is going to be the lumen of my intestine. Right, lumen just means that if here's my intestine, lumen is the hollow space where all the digestive processes are happening, the breakdown is happening. This is where all the food is getting smooshed together with all the enzymes and getting broken apart. We have our simple columnar epithelium, which are kind of making this line with this lining. And what's on the other side? So we have inside here is where all the food's breaking down. What's on the other side of this epithelium? What do we do with the nutrients that are the results of digestion?
They get absorbed.
They get absorbed. Absorbed where? How do we absorb things?
Through the microvilli.
So that those are going to help increase the surface area. But ultimately, what takes up the nutrients and takes them where they need to go?
The capillaries.
The capillaries, exactly. So on the other side of this epithelium are the capillaries, the bloodstream, which are going to pick up the nutrients, the sugars and stuff, and deliver them. So kind of just trying to give you some context here. We're looking kind of right at this place. We're looking at the lumen, and we're looking at the epithelium, and then we're looking on the other side where the blood vessels are that are going to pick up the nutrients that are the results of the breakdown. So that's why I'm saying lumen. This is the inside. So here, I've got like little glucoses. These are going to be my little glucoses. These are the result of the breakdown of my food into the basic biomolecules. We'll look at that in way more detail. So here, glucoses. And again, on the other side are my capillaries. So I have the glucose up here in the lumen. I have the simple columnar epithelium making this boundary between the lumen and the inside of my body where the blood vessel, the capillary is. I need now to figure out how am I going to get the glucose from here into here. So that's transepithelial transport. I'm crossing across an epithelium. I need to bring the glucose into here. And it's going to use all the things we've been talking about. So let's see how this all comes together and works. One thing is we are going to have the glucose coming in using a symport that I've already kind of talked about. That couples the glucose to the movement of sodium. So glucose only comes in if there's sodium. It's going to be driven by a sodium gradient. That is, what do we call that? If I'm moving glucose in to the cell using the power stored in a sodium gradient.
Secondary
Secondary
transport.
transport.
So this is going to be an example of secondary active. In order for this to work, I have had to previously set up the sodium gradient. That's going to be done by primary active transport. So I've got an active pumping of sodium out of the cell. And that's only going to happen using the power of ATP. So what do I call this if I'm actively pumping sodium out using directly the power of ATP?
Primary.
Yeah, so this is going to be an example of primary active transport. All right, so we are getting a whole bunch of glucose now into the cell because we are using the secondary active transport. On the other side, there's not a lot of glucose because the glucose, all the glucose that ends up on this side is getting taken up into the capillary. So for the glucose to move out of the cell onto the capillary side, it's just passive. It will just move along its concentration gradient, but glucose can't cross the membrane on its own. What does it need to cross the membrane?
Sodium.
A carrier.
It needs a carrier, exactly. So we're going to have some glucose carrier.
Can I ask a quick question?
Sure.
So it's secondary transport to get through the columnar epithelium, and then it's primary transport to get it into the capillaries?
No. So let me finish this and then see if the question still makes sense.
Okay.
So we have this carrier, this glucose transporter. What do I call this? If the glucose is just moving along its concentration gradient, but it requires a carrier to move across the membrane.
Passive facilitated diffusion.
Yeah, it's facilitated diffusion. Is that active or passive?
Passive.
Passive, that's passive. That's just glucose moving from high to low concentration, but needing the services of a glucose carrier, a transport or protein to cross the membrane. So what we have here, glucose in the lumen, to actually go from the lumen into the cell where there's already a lot of glucose, we're going to need an active transport to pull the glucose into the cell. That's going to be done by the secondary active transport using the sodium gradient and this symport that couples sodium and glucose movement. The reason this can work is because we set up a sodium gradient, because we have an active transport where we're always pumping sodium out of the cell. So we're always keeping the concentration of sodium very low in the cell, so sodium wants to come in. That's why this secondary active transport is working in the first place. And then if we're looking at, so sodium, glucose is entering using secondary active, but to leave the other side, the capillary side, it doesn't take any extra energy. We've got a concentration gradient on our side. The glucose wants to leave out the backside here, but it just needs a doorway. It needs that glucose carrier in order to make it across the membrane. So this is showing examples of active and passive, primary active, secondary active, and also just illustrating this idea of transepithelial transport, which we will actually see. We'll see in the digestive system, we'll see in the urinary system, we'll see lots of places in the cardiovascular system. There's lots of places where you have some epithelial sheet and you want to intentionally move something from one side to the other side. And it's usually accomplished, using different membrane transport proteins in some system kind of like this.
And is this example, the sodium creating the concentration gradient for the second active transport? It can be, is it always sodium or it can be any, it depends on where in the body it's taking place.
Right, this is just an example. This is an example. It can be different things. This is the same system that your kidney tubules use to reclaim glucose from the filtrate during filtration in the nephrons. I talked about why the rate limit of facilitated diffusion was important to pay attention to. How fast I can move glucose from here to here is going to be limited because we're using these carriers, which are limited in how the maximum rate things can move across. So like when you have diabetes and you have way too much glucose that ends up in the filtrate to try to move back into the bloodstream, and you overwhelm the ability of these carriers to do their thing, that's when you start getting like sugar in your pee. So, again, it's similar and different. It'll make sense when we get to the kidneys, but the idea that the facilitated diffusion is rate limited means there's a maximum rate that we can actually move things across in transepithelial transport. And if the body is functioning in its normal range, it works fine. But if things are going wrong, you can actually hit a wall where you actually realize like the system can no longer function properly because it has a limited capacity to how fast it can move things. Okay.
I have a question.
Uh-huh.
So glucose comes into and then ultimately out of the other side of the intestinal epithelium. And when it goes and gets picked up by the capillary, it has to cross a thinner sheet of epithelial
that's
cells.
another transepithelial transport to get into the capillary.
And is that facilitated diffusion at that point?
Let me think about that. I think it's probably going to be a little more active as well because you normally have quite a bit of sugar in your blood. I'd have to look. There's the movement of things from into the capillary. There's lots of different options, and I don't remember whether it's an active or a passive thing for the glucose. So I would say I'm not sure. Maybe or maybe not.
Okay.
All right. So other comments, questions about this transepithelial transport? Again, this should all make sense because it illustrates important types of transport and illustrates important transport proteins. It illustrates also this idea of moving things across epithelia, which is going to come back in a variety of forms as we go through the semester. All right. So what are other functions of, so we're doing membrane proteins. So transport is a big one. Another one I'll bring in just because it's going to be important to understand transepithelial transport. There's cell-to-cell attachment. So again, you're made up of like 10 trillion cells, and most of them are kind of stuck together to hold their basic form. Some of the cells just stick together by these like proteoglycans, these kind of cellular glues and stuff like that. But where there's particular stresses or there's particular conditions that make it important, you can have special proteins that make specialized attachments between different cells. So for instance, one kind of cell-to-cell attachment are called tight junctions. Tight junctions are kind of like Ziplocs that actually kind of create this. Think about a Ziploc. A Ziploc, you have two sides of a Ziploc, but they kind of interleave in this way where, water can't even get through, right? So it's still two sides of the bag, but they have the tongue and groove have gotten into each other. So it makes this kind of waterproof seal. Tight junctions are where you want to make sure there's this really super tight seal between the cells so things can't wiggle through. For instance, in transepithelial transport that we just saw, it's important that no sodium or glucose are just kind of cheating and wiggling in between the spaces in between the epithelial cells. So in order to make sure that doesn't happen, we're going to have tight junctions. So I can give it a. That means if something is trying to move across the membrane, boom, it can't. So having the tight junctions ensures that only the things that we were intentionally moving from one side to the other through these specialized membrane proteins are moving. And even though there's all sorts of gradients and things that things want to move, they can only move if they're being intentionally moved. They are not going to be able to just kind of wiggle and sneak through the spaces in between the cells. But does that make sense? So tight junctions are important, particularly in these epithelial sheets where you're trying to really control what can move from one side to the other side of the layer. Another kind that is going to be important is where you have cells that are under a lot of physical stress. Like, again, if you've been in anatomy, you've learned about skin cells with all the little desmosomes, right? So desmosomes, we have cells, and I don't want them to pull apart. They're under a lot of physical stress. They might be the epidermis, or maybe it's actually your cardiac muscle cells. Your cardiac muscle cells are under a lot of stress as the ventricles squeeze with all this pressure to push your blood around. There's a lot of stress on these individual cardiac muscle cells that could pull them apart. And the last thing you want is your wall of your heart to rip under pressure, right? So we have what are called desmosomes, which I like to think of as kind of little rivets and cables. Oh, come on. There we go. So desmosomes, again, they're like little rivets and cables. So there's proteins that connect one cell to another in a very strong way. And then there's usually different protein filaments, that cables that I'm talking about, that kind of span and connect things within the cell as well to kind of stabilize these rivets to the cytoskeleton in the cell. So it's really hard to pull these cells apart, even under a lot of tension. Again, we're going to find this in a number of places where you don't want cells to rip apart. Again, in anatomy, it's important in like the epidermis, but in our class, it's holding like cardiac muscle cells together so they don't rip apart. So desmosomes The last kind of cell-to-cell attachment you need to know are the gap junctions. So here I have cells. And I'm going to look at, they're called gap junctions. Gap junctions connect the cytoplasm. So they're like little they call them like connexons. So one cell is just continuous at some level with the other cell in terms of cytoplasm. Things can drift from one cell to another. For our class, the main physiological impact of cells having gap junctions is that ions can easily drift from one cell to another. And that means that the cells are electrically connected. If ions can move from one to another, we talked about ions moving charge being the electric current. This is going to electrically connect the cells. So that is going to be important when we get to smooth muscle, cardiac muscle. You excite one, you excite all its neighbors. They're all electrically connected. There's also going to be some gap junctions in the neurons as well, although we're going to see more synaptic transmission rather than electrical synapses in that case. But whenever you see gap junctions, you should think about electrical connections. If you electrically excite one cell, all the cells that are electrically connected are going to be excited as well, because they're all kind of like little wires connecting them all together. So these are the cell-to-cell attachments that you need to know about. All right. So membrane proteins, we have transport, check. Cell-to-cell attachment, check. But wait, there's lots more. What are some more examples here? Different jobs membrane proteins do.
They could act as receptors for hormones.
Yeah, receptors for hormones or for neurotransmitters, or even for just environmental ligands. Like I said, like the taste buds are going to have receptors that just recognize sugar. And that's how your taste buds work, is having some membrane protein that can bind and recognize and bind sugar and then create some response that you experience as tasting sweetness. So let's talk about receptors. So this is again, membrane proteins continued. And actually, you know what I'm going to do in order to make our life, I think, make things flow better? We are going to talk a bunch about receptors. We're going to talk about different kinds of receptors, the details of how receptors actually result in actual actions within the cells, and ideas of affinity and agonists and antagonists and all sorts of stuff. So we are going to spend a lot of time looking at receptors. I want to just finish our list of membrane proteins, because the other things aren't going to require an involved deep dive. So just to kind of finish our thing, we talked about transport. We talked about cell-to-cell attachment. We're going to talk about receptors in a lot of detail. Let's just kind of finish our list of membrane protein jobs. What other things do membrane proteins do?
They could recognize invaders.
Or I would say, yeah, they give your immune system the ability to recognize your own cells versus invaders. So like cell identification. You know, we're going to be spending much more detail, like talking about major histocompatibility complex, MHC proteins in the membrane. It's kind of like the barcode for a cell that belongs to you versus somebody else. So there's membrane proteins that tell your immune system this cell belongs here versus this cell does not belong here. So we'll talk much more about how all that works when we get to the immune system. So cell ID, though, is important. But wait, there's more. Cytoskeletal attachment. That's going to be important. You know, cells hold their shape, right? Some cells look like that, some look like that, some look like that. You know, that means something inside is scaffolding and has to anchor the thing and make it hold its shape. When you have endo and exocytosis, things are pushing and pulling and moving. That's got to be something from the cytoskeleton attached to the membrane. So cytoskeletal attachment, and when we get to villi or microvilli and cilia and stuff, we're going to see cytoskeletal attachment.
Would transduction be one?
So that's going to be related to the receptors, I would say.
Okay.
When we get to the receptors in more detail, we're going to meet a whole family of special proteins, all the G proteins and the like adenylyl cyclase, things that are creating the second messengers that are going to be part of signal transduction. But I would say, to simplify things, let's just kind of include them in this category of receptors. All right, so membrane proteins. Cell ID, cell cytoskeletal attachment, but receptors are what we need to spend some more time in. You know, I know we spent quite a bit of time doing the tutoring stuff, so it's already 9:30. So maybe we should take a break. But I do want to spend a little more time talking about receptors before we are done with this. What we're going to do now is dig deeper into receptors, because they're kind of like the eyes and the ears of the cell. These are how cells can detect what's going on around them. It's how cells can, you know, give messages, find out if other cells are sending messages to them. It's also how they can detect just the environmental conditions around them. So let's talk about receptors. So let's say this is my cell. You know, receptors are specialized membrane proteins. You know, and they have some kind of a binding site on them. You know that binds a specific ligand. Again, we've used that word before, ligand meaning a signaling molecule. You know, the classic analogy people use is the lock and the key. You know, so I, you know, have some ligand that perfectly complements the binding site of the receptor. So some receptors are going to be specialized to bind a neurotransmitter, let's say serotonin. Some are going to be specialized to bind a hormone like insulin. Some are going to be specialized to bind maybe just environmental things, like I said, glucose, if you're trying to monitor blood glucose or detect glucose in your saliva as part of your taste buds. So the ligand is just a molecule. Binding is this kind of lock and key, but again, you're not breaking the key off in the lock, right? It binds, it holds in, it has an effect, and then just due to all of the energy, it releases and drifts away again. So, right, a lock to your home is going to be useless if every time you unlocked it, you broke the key off and nobody else could use it again. You use it and then you take the key out, and then it's ready to go again for another round sometime later. Next week, I'll get a little deeper into what exactly is binding. I'll give you like specific examples. I have a nice little presentation showing what exactly does it mean if a serotonin binds a serotonin receptor? How does it fit in physically? How does it make all the appropriate chemical bonds, little hydrogen bonds here or ionic bonds there? But for right now, you can just think of binding as kind of the key fitting into the lock. So let's leave it there. There's the idea of endogenous ligands. Endo, within, generated, created within. These are the naturally occurring ligands like within your body. These would be like serotonin or insulin.
I couldn't hear what you said.
So endogenous just means the naturally occurring things that are in your body. As opposed exogenous ligands. A lot of like pharmaceutical and recreational drugs function as exogenous ligands. The basic idea here, exogenous means from without. So something that you took or you smoked or you got injected into you or whatever, somehow this molecule got into your body, but it is acting as a ligand. It is getting in and it is binding the receptor and having an effect there. So these are, from outside your body. Typically, they have to look awfully like the ligand. So they kind of mimic the endogenous ligand. Again, I will show you next week examples like, if you eat magic mushrooms, why does psilocybin make you trip? We're going to see it has a very similar structural and functional similarity to serotonin. And it can get in and bind on a serotonin receptor. So if you have asthma and you're sucking down albuterol, for your asthma attack, albuterol has a very similar shape and properties that it can bind in adrenaline receptors and help dilate your bronchial passages. Morphine or fentanyl or any of those pain, kind of pain meds are going to have the ability to get in and bind on your naturally occurring opioid receptors and turn on these pain mediating pathways. So exogenous ligands, they're from outside, but they're close enough to the naturally occurring or endogenous ligands that they can get in and they can bind at the receptor. The thing that's interesting though is endogenous ligands, they bind, but sometimes they bind and they actually activate. So let's talk about exogenous ligands. Whoops. If they bind and activate the receptor, we call that an agonist. We'd say like albuterol is an agonist to the adrenaline receptor because it binds the adrenaline receptor and it activates it and turns it on like, kind of just adrenaline within your body would have. That's what dilates your bronchial passages, makes it easier to breathe. Again, morphine also, it can bind these opioid receptors and activate them, and reduce your perception of pain. There's another thing that can happen though. It can bind and get in there, but not fit it just right and actually not activate it. So the other possibility, it can bind and not activate receptor. But because it's binding, but it does block, other ligands from binding. So this is going to be called an antagonist. Like for instance, I use the example, albuterol is a agonist that turns on the adrenaline receptor, which is useful if you're trying to like breathe, if you have asthma. There are also antagonists to the adrenaline receptors that you can take, like the beta blockers. The beta blockers get in there and they bind the adrenaline receptor, but they don't turn it on, and they keep the naturally occurring adrenaline and from actually binding and turning it on. So those would be useful for like controlling blood pressure because they keep your heart from speeding up. They actually, people take them for performance anxiety because you get scared and adrenaline gets released into your body, but it can't actually get to the receptors. So you don't actually get that butterflies in your stomach because your heart's not speeding up things, so an antagonist blocks the receptor. Does that make sense? All right. So agonists get in there, bind, and they activate it. Antagonist gets in there, binds, but it doesn't activate it. It actually doesn't turn it on, but it blocks other things from getting in there. There are also things in the middle, things we call partial agonists that turn them on, but not quite as strongly as an endogenous ligand would. So there's a continuum that gets more complicated. The other thing we can talk about exogenous ligands is affinity, how tightly it binds. So if I have some cell and some receptor, and I could have like a low affinity ligand. What that means is it gets in there, but it breaks apart. Gets in there, hangs out a little bit, then it goes out. Gets in there, hangs out a little bit, and then it breaks off. So low affinity means it gets in there, but it doesn't hold on that tightly, and it tends to leave pretty quickly. High affinity is it binds really tightly. High affinity, it goes in there and it really stays, and it's boom, boom, and eventually it has to break off, or else, it's like breaking the key off in the lock. But high affinity is going to spend a lot more time connected to the receptor and having an effect there. This can make a big difference in the dosages of something, right? So let's think about opioid receptors, like the receptors that are part of turning on these pain mediating pathways. So I can think about, what's a classic, if you're trying to like knock down pain in somebody, they get wheeled into the ER. Ah! What's something you're gonna like kind of shoot them up with?
Morphine.
Morphine. So think about morphine. You need quite a bit of morphine to really make somebody comfortable if they're in a lot of pain. Maybe like 30 milligrams or something you're gonna shoot into them. What's another really common opiate that gets used?
Oxycontin, oxy.
So there's OxyContin. So that's I'm going to put in fentanyl here.
What is the D one? It starts with a D.
Dilaudid.
Dilaudid. They use that a lot too.
Yeah, Dilaudid you need a lot less. So Dilaudid you'd need like a couple of milligrams. Fentanyl, I'm putting fentanyl here because you need really little, lot less. You need about 100 micrograms. Right, that's 0.1 milligrams. So that's 300 times less than this in order to have, the same amount of kind of pain suppression. And part of this is thinking about affinity, right? If one of these gets in there, it can hold on and have an effect for much longer. So you don't need as much floating around in order to have a strong effect. If you have something with lower affinity, you're going to need more of it floating around because it gets in, it gets out, it gets in, it gets out. So you need more of it just floating around to have the same ultimate effect at the receptor. If you have something that has very high affinity, it gets in there and it holds on, and you don't need to have as much floating around. Fentanyl is particularly, it's tied into all sorts of problems. It's used clinically all the time. It's very useful. But because it's so effective at such tiny doses, it's really easy to overdose on it, particularly if you don't know what you're taking. It's used to adulterate a lot of street drugs because it's cheap and easy to make, and you add a little bit and then the drug is like, whoa, this is doing something. I've got my money's worth or something. But then a lot of people don't realize they've got this high potency opiate on board, and then they overdose and die. So yeah, be really careful with street drugs. I just had a friend's son who was 24 and died, who just thought he was going to have fun. So again, fentanyl in and of itself is not evil. It's like, but people adulterating things with it or using it and not knowing how much they're using it in what context is very bad. So affinity. We can tie together talking about like opiate overdoses. Too much, if you have too high of a level of opiate drugs in your body, you're gonna like stop breathing and die. So it's not good. So somebody is overdosed on, who knows, fentanyl or morphine or heroin or any of these. They all are exogenous ligands that are binding like the little opioid receptor. What is the first order of business if somebody is presenting with an opiate overdose?
Foaming of the mouth,
Well, how are you going to try to keep them alive?
Narcan.
Narcan. Let's talk about Narcan. How Narcan is a nice poster child for a lot of these things we're talking about. And everybody, if you don't know what Narcan is, you should know, because it's a really easy way to save people's lives if they're overdosed. It's naloxone is its official name, but it is a very high affinity opioid antagonist. So why is this going to be helpful? Somebody just took too much heroin or morphine or whatever, and they are slowing down and they're going to stop breathing and they're going to die if we don't do something here. Why is giving them a high affinity opioid antagonist going to be a good thing?
Because it's going to bind and stay, so it can't open up to have access for another thing to connect.
Exactly. So let's say this is my little receptor. And let's say we got all these like kind of heroin molecules all floating around that have been getting in here, and there's too much. It's not just like a nice floaty high. This person's going to stop breathing and die if this keeps going on. Then you get Narcan. Narcan, once it gets in there, it holds on. You know, so these little heroines, they can try to get in there, but as they get in, they bump out, and then the Narcan gets in, it doesn't let go. And then these guys are, it's not like these guys are gone, they're floating around, but they can't get access to the receptor. And so being high affinity, it basically kind of noses out the other opiates that are floating around. And it's an antagonist, which means that, yes, it's binding, but it's not actually activating the receptor. So all of a sudden, we don't have activation at that receptor.
So the Narcan causes inactivation.
Correct. Right. In fact, a lot of times I've talked to people who've administered Narcan to people, and the people often get incredibly pissed because they were just like gone in this warm, fuzzy, floaty place, and all of a sudden they're back, like kind of almost instantly, just back there in this kind of, and they're like, what the heck did you just do to me? It's like you're saving their life, but you kind of pull them out of their high, because as soon as this Narcan grabs in, you know, basically the high is over.
What happens to the heroin molecule? Heroin molecule.
So eventually they get cleared, just like any other thing in your body. There's different enzymatic pathways in your liver that kind of break stuff down. Your kidneys are removing stuff. So it's kind of the same way. If you like, you know, do a bunch of tequila shots and get really drunk, eventually you're not drunk anymore because the alcohol clears out of your body. You know, these guys are floating around and they're being cleared eventually at some certain half-life. And then you just have to make sure you keep the Narcan levels up high enough until those little heroin molecules are cleared out and are no longer a problem.
So if the heroin molecules are actually bound to the receptors, does the Narcan come in and kick them out?
Yeah, and like I said, but being bound is a temporary thing. So you're bound, but then you kind of let go. So when they let go, the Narcan goes in there and then it holds on much tighter. So it's much less likely to let go. So it doesn't give the heroin a chance to get back in there. So
I have a question. So in this case, would the Narcan be like the substrate? Because if the receptor, that would be, I'm just have a question. Are these, it's like enzymes?
Ligand. No, so it's a, I'm glad you asked that. People get confused. Enzymes versus receptors. They're similar in that, both like kind of bind something like a lock and key. So I think that's why people kind of get confused, because both of them, binding is this kind of critical part, but kind of the context and what they're doing is pretty different. Enzymes, bind substrates, to catalyze chemical reactions. Receptors, these are membrane proteins. Actually, not all, it's, that bind a ligand. That is a signal to the cell. Right, so they both bind. In both cases, the shape actually is important for binding, but kind of the context and what they're doing, the role they're playing is very different. A receptor is kind of sensing what's going on. It gets this message and then something happens as a result of getting the message. So these are like kind of get messages. Enzymes like make reactions happen. And even the word we use for what they bind, we say the enzymes bind the substrate. That's like the chemical or the molecule that's attaching to the enzyme that is going to then be catalyzed. Receptors, we say bind the ligand, like the signaling molecule. That's the most common way to use the word ligand, kind of a signaling molecule. Which then kind of actually detaches and is ready to go again, usually. The substrate usually is going to be irrevocably transformed as a result of whatever chemical reaction is happening by the enzyme.
I'm sorry, so you said that the substrate is going to be, I understand the substrate's going to be different, but the ligand, is that something that can be reused?
Right, kind of back in, right, if in this case, the ligand, it could be this heroin, it could be anything, it could be Valium, it could be just like a benzodiazepine. It's just some molecule that's in your body and is floating around, and it binds and it lets go, and it binds and it lets go. But the idea is not to have the receptor catalyze some reaction. It's more, it gets the message and then there's something that happens downstream as a result of getting that message.
Okay, thank you. And then as far as like, you said that like, obviously when you do heroin, it doesn't last forever. So when, I think someone had brought this up before, but when your high is essentially over, where does that heroin go? Does it just get flushed out of your system via like your kidneys?
It's partly kidneys.
Your liver?
Yeah, liver. There's a lot. We'll talk more about the liver later, but the liver has all these enzyme systems whose job is to kind of degrade and break things down. So that's a big part of what the liver, the detoxifying of the liver is having all these enzymes that, that's a big part of pharmacology, is kind of understanding what happens to these different molecules and drugs once they get into your body and how they get broken down, or sometimes they get broken down into secondary metabolites, which sometimes just get removed or sometimes actually might be active on their own as well. But yeah, that's something we'll touch on in this class. But when you get further on in your studies and get in deeper into pharmacology, that's where you really get into depth with what happens to those things after they've kind of done their thing, or how your body clears things.
Okay, thank you.
Sure.
I have a quick question.
Uh-huh.
How does tolerance level play into this? What happens to the receptors when people build up tolerance to drugs?
So a big part of that can just be down regulation of the how many receptors you've got. So let me get back. Here, let me put a new page. Here's my cell. Let's say I have a whole bunch of receptors, a certain number of receptors for the same thing. And, let's say I'm on maintenance, there's people who are, take morphine every day or take oxycodone every day or whatever. And so you have a lot of this stuff and the cell starts, it's kind of like if the volume on the stereo was always loud, and you just realize like, man, the volume is always loud. I'm going to just stick cotton in my ears to try to make it more normal. So you actually get the cells too far down regulating how many receptors there are. So now there's not as many receptors. So it's like, it doesn't hear the messages loudly. It's kind of adjusted for the fact that there's more of the ligand floating around than normal. I mean, what that means is to have the same effect, you need to have even more of the ligand because there's less receptors for it to be interacting with. And with the opiates, whether it's morphine, like you can buy little pills that are like 100 mgs of morphine that I'm assuming, and most anybody here, if you took one of those, you're dead. But if you are on maintenance morphine, that is kind of what you need to maintain an effect, the pain, pushing the pain away, because you've had that tolerance. And so you need to have very high doses in order to actually have an effect. And I think a lot of that is from the down regulation of how many receptors are actually available for the drug to bind to. Psychedelics, if you drop acid one day and you take the same, like normally like about 100 micrograms of LSD would be enough to trip pretty hard. If the next day you took that same amount, you'd barely feel it. For the same basic idea that your cells are like, whoa, the volume was really high. We're gonna like back off the number of receptors. I talked about how cells can constantly adjust their membrane properties. So it takes a few days before this dose is going to work again. You have to kind of let everything kind of go back up to its more normal levels. So, there are different things that happen with tolerance, but just one of the simple things that happens is just down regulating receptor density. So you need more of the drug in the system to have the same effect because there's less locks for the keys to fit into. So questions about agonist, antagonist, affinity?
Sorry, I just sort of a question-ish. Would it be fair to say that like the ligand binding to the receptor is like a precursor to a reaction? Like how you were comparing it to the enzyme, like that's like that's something that happens further down, or that doesn't even, or it's just not related. Like it doesn't work.
I mean, I think your idea is correct in that the ligand binding the receptor kind of starts a process that results in something happening, but using that word precursor, I think makes things confusing.
Okay.
So I wouldn't use that word, but the basic idea is correct. That, in fact, I don't,
because the receptor and the ligand itself is not like forming any sort of reaction.
Sometimes it does. We're going to see there are like directly gated channels. Like if In your muscles, you have the nicotinic cholinergic receptors, which are gated channels that are that work by the binding directly. So like, for instance, I'm going to talk about these in more detail later. So here I have a cation channel, and it's got a little gate. So it can be open or closed. And then I also have a binding site for acetylcholine. So this is one big daddy rabbit protein that has everything all kind of in one. And if acetylcholine comes in and binds, it basically opens the gate and lets the sodium through. So in this case, there's not like there's not like lots of steps in between. It's directly, basically the acetylcholine binds there. There's some conformational change that opens the gate and sodium rushes in. You know, that's really different from another kind of, maybe over here, I'm going to put another one. I'm going to put, this is going to be a muscarinic cholinergic receptor. Which works really different. In this case, and we're going to talk about this in more detail later. In this case, if acetylcholine binds onto it, it turns on a G protein, which turns on adenylyl cyclase, which makes cyclic AMP, which activates protein kinase, which phosphorylates some protein, which then goes and finally opens some channel. You know, so this is what we, you know, indirect, you know, second messenger mediated signal transduction.
But either way, it's like triggering something, whether it's like direct or indirect.
Yeah, because otherwise, what's the point? I mean, the message comes in, unless something happens, binding in and of itself is like, you know, the binding has to have some result. If the binding doesn't result in something, then what's the point of it? It's just like something sticks and falls off. Nothing happened. So the binding of the ligand to the receptor opens some channels maybe for ions to move, or maybe it turns on some enzyme to turn on some metabolic pathways. It makes something happen. So does that make sense?
Yes, thank you.
Yeah, so, but the word precursor sounds more like there's a molecule that then gets turned into another molecule. So that's why I don't want you to use that word, because in this context, people would misunderstand what you mean with that word precursor. But yeah, on Tuesday, we'll talk much more about, you know, there's not much more to say about directly gated. You kind of get it. It's like it happens, you bind and it automatically opens the gate. But these signal transduction using second messengers, it's really important that we understand that. And so we're going to look at that in more detail on Tuesday. Yeah, so we'll look at in way more detail. You know, in fact, some drugs, I talked about a lot of drugs work as these exogenous ligands, you know, things like the benzodiazepines. If you're taking Valium or Xanax and you're kind of getting smoothed out. It's just because you're binding these receptors that would normally bind GABA, which opens up chloride channels, lets more of this anion into the cells and makes them more negative and less excitable. Or if you're smoking pot, you know, THC is going to bind all of the like cannabinoid receptors and turn those on, and you're going to get high. So a lot of both recreational and pharmaceutical drugs work by binding, you know, again, mimicking. They have to look close enough to the naturally occurring ligand that they bind the receptor and hold on, and then they can either activate or block it. But then other drugs, like another super popular drug like Viagra, which, you know, for like facilitating erections, it actually works within the second messenger system. It actually messes with levels of second messenger cyclic AM or cyclic GMP in the cell that keep, you know, we talked about how there's the receptor binding and something happens, and that there's a bunch of stuff happening that makes it happen. Like Viagra works in this pathway here. So if you turn this on, that once the pathway starts, it doesn't turn off. So the guy gets an erection and the erection persists because you have changed the system so this part lasts a lot longer than it would have normally. So some drugs actually work downstream from the receptor, but it's still part of that process. And we'll talk more, we'll talk in detail on Tuesday, like why Viagra works, because it's actually useful in understanding the second messenger pathways. So comments, questions about any of that? Okay.